So thanks for having me. It's a pleasure being at this workshop. Obviously, we're talking about things in the larger rubric of climate and health. And I wanted to give some of the sort of my philosophical thinkings about the ideas of climate and health, climate and infectious disease in particular. And I just wanted to put forth the notion that, climate and disease is not a new notion to a certain extent, okay? This is the oldest reference I've ever come across to malaria. If you make it all the way to the 7th book of the history of the Peloponnesian War by Thucydides, you'll see him talk about the destruction of the Athenian expedition at Syracuse in 413 BC, and he wrote this in about 406 BC. And he says, and I'll just read it, after a particularly devastating battle, the Athenian generals consulted upon the disaster which had happened and upon the general weakness of the army. They saw themselves unsuccessful in their enterprises, and the soldiers disgusted with their stay, disease being rife among them, owing to its being the sickly season of the year, and to the marshy and unhealthy nature of the spot in which they were encamped. And in that last clause right there, he's describing malaria. He doesn't know what it is. He doesn't know how it's transmitted. There's no germ theory. They don't know that mosquitoes are the vector of it. But he's pinpointing the fact that it was recognized that marshy areas were unhealthy, and at particular times of the year in particular, summer in Sicily, it was particularly unhealthy. So there's a recognition that human health and disease were inextricably linked to the environment, including the climate, going back almost 2,500 years now. If you were to run forward into the 16th century, 16th, 17th, 18th centuries in Europe, obviously there's the field of medical geography, and a lot of what they were trying to do there was to sort of catalog and understand what's going on with the patterns of disease. And there was an understanding that there were certain waters and certain airs that were healthful, and there were less that were less healthy. And there's a rich vocabulary in the English language, and I'm sure other European languages describing these different types of things. And the notion of the spa sort of came back into vogue, that people would go certain areas to get to more healthful climates, places where it was just a better place to live. So now, of course, we have this increasing attention on the effects of climate variability and global warming on human health and what's going to happen. And the picture, of course, is variable as to what's going to happen in the future. It's not a single monolithic change in temperature of the hydrologic cycle, for instance. And one of the things that people look at, and one of the issues that we have to deal with, is trying to look at it beyond just the broadest terms. And I'm not trying to disparage anything here. I'm actually going to be echoing some things that Mercedes was talking about earlier. But if you were to look at what we have here on the left, what you have is a picture of the increase of global average temperatures over a 125-year period. And you see a rise in that. You also have a picture right here, I'll use the pointer, of the distribution of malaria as it currently stands. Upper right there, that's the Anopheles gambii mosquito, which is the principal vector of malaria in sub-Saharan Africa, where 90% of malaria transmission occurs. Now, that mosquito has an optimal temperature range and tolerance that's been fairly well established in laboratory studies. It's got a window of minimum and maximum temperatures at which it thrives. If it gets warmer but not too warm, it's going to complete its extrinsic incubation cycle very well for the pathogen within it. It's also going to complete its gonotrophic cycle and lay its eggs. It's going to sort of accelerate the whole process by which it goes through biting, acquiring pathogen, laying its eggs, and then seeking another host meal and potentially transmitting to the next person. So one of the things people thought about initially was, well, if global temperatures are warming, and if Europe and Asia and North America become more and more like what it's like in sub-Saharan Africa, the Anopheles gambii mosquito could get on a plane, come off, and find that New Haven, Connecticut is pretty hospitable in 100 years, and then it's going to set up camp and it's going to go around and do its business. Another thing people have looked at initially, Mercedes obviously showed much more detailed and elegant work on this, was looking at the fact that there's association between El Nino and what's going on with cholera outbreaks. And the thing is about it is that when we talk about climate, and we think about it at very long time scales. And we typically, though we don't need to, think about it at very large spatial scales. But the fact of the matter is, the spatial scales at which these diseases take place, the spatial scales that is experienced by an infectious disease, the perceptual scale of a vector of that disease, like a mosquito, is very small. They have a very small world they live in. And if we're going to understand what's going on at that, we have to marry across those scales. And we have to start thinking about things beyond large-scale patterns of change and look at more localized hydrologic and meteorological variability. And my contention is that if we can understand that at a more localized scale, we can then build up to it and then factor in those projections and figure out what's going on. So To think about it again, if we just take this example of malaria once more, we would not need to just think about this global pattern of warming and what that's going to do to temperatures across broad swaths of continents. We have to think about the local rainfall. We have to think about the local temperature changes, the local humidity changes, and the local environment. And the other thing that we have to do, and again this echoes what Mercedes says, is we can't think about climate and health in a bubble. We have to think about all the other factors that actually impinge on and affect and modulate rates of disease transmission. We need to think about the biology. We need to think about the genetics. We need to think about the medical and public health infrastructure, land use, socioeconomic conditions. We have 8 species of the genus Anopheles here in North America that can transmit malaria, but we don't have malaria anymore. It's because we drained our swamps, we managed those resources, we paved over tons of areas, we build constructions and buildings like this with air-conditioned systems and good screens and whatnot that keep insects out of them. We husband our animals in other places for the most part, aside from our pets. And so we've really segregated ourselves from the mosquitoes. And when we do that, we cut off the transmission cycle that is vital for the sustenance and the sustained transmission of a pathogen such as malaria. By all accounts, Houston should be a malaria zone, but people go from their air-conditioned house to their air-conditioned car to their air-conditioned workplace. And when they see a mosquito, they swat it. So you would need a breakdown in that socioeconomic conditions, the medical and the public health infrastructure, I would argue, before you would actually see some effect of temperature necessarily manifesting as a change in malaria rates by the introduction of a mosquito. So we have to think about it in that larger context. All right. So off my soapbox, and I'm going to talk about West Nile virus a bit. So West Nile virus is a mosquito-borne disease. It's a flavivirus that's similar to Japanese encephalitis virus. In the United States at this point, we're getting up to 10,000 clinical cases per year, which manifest as meningitis, encephalitis, and a more milder form, something that's called West Nile fever, something that doesn't penetrate into the meninge across the blood-brain barrier. Now, the majority of the cases, though, are not actually documented clinically because they're mild or subclinical and people don't actually seek medical attention for it. So there are a lot more cases per year than that 10,000 would indicate. Okay? At present, there's no vaccine available for humans. There's an equine vaccine out there, but the CDC does not recommend that you go out and buy it and take it. The West Nile virus transmission cycle is summarized in the schematic right here. And what it consists of is this zoonotic loop right here of transmission between Culex genus mosquitoes that are vectors and typically avian hosts. So what happens is that a mosquito comes along, it bites an infected host that has a high viremia, takes on the blood meal, it gets infected with the virus. After a certain period of time that's referred to as the extrinsic incubation period, the virus has multiplied within the mosquito host and has moved to the salivary glands. If the mosquito survives, lays its eggs, she can then go on and seek another host meal, which you'll use the blood for to feed the next batch of eggs. And in doing so, when she feeds on another bird, she can pass that virus on to the bird. So we have this transmission cycle going on in North America since the introduction of West Nile virus. And because of this, we're never really going to get rid of the virus. We're never going to keep birds and mosquitoes apart. So it's something that we're going to have to deal with for a long time, I would suspect. Now, in addition, we have these things right here. This is sometimes referred to as spillover transmission or incidental infection, and we're sometimes referred to as dead-end hosts. And these are other organisms, such as people and horses, that can be infected with West Nile virus. So should one of these infected mosquitoes bite us, we can become very sick from it in some instances. Now, typically, we don't run a high enough viremia to pass it back to a mosquito. So we're not participating in this transmission loop that sustains it within the environment. Hence, we're sometimes called a dead-end host. So West Nile virus first appeared in North America in 1999 in New York City. And that year, there were 62 cases of West Nile virus, all within the New York State. And there was a little bit of evidence in avian, animal, and mosquito infections in some surrounding states in that first year. So one of the questions at that point was, well, how quickly was it going to spread? And as we found out, it spread fairly quickly across the whole continent. So this is what the picture looked like in 2000. You can see it spread up down the northeast corridor a bit more, but still, you're not getting too many human infections. Here's 2001. It spread all the way past the Mississippi River. It's saturated the Mississippi River Valley, essentially. It's all the way down to Florida. Here's 2002. It's made it all the way to the West Coast. And then 2003, have some big outbreaks, almost 3,000 cases in Colorado, almost 2,000 cases in Nebraska. and it's basically saturated the whole country at this point. There's 2004. If you pull up any more of the CDC maps since then, it's pervasive throughout the country. So here's some plots for 8 of the states, California, Colorado, Florida, Idaho, et cetera, showing you the pattern through time in total number of cases of West Nile virus fever, encephalitis, or meningitis, as reported through the CDC on an annual basis. And what you can see is that there's sort of some evidence that in the first year or two, sometimes it's 3, sometimes it's a little more, after introduction, there's a large outbreak initially, a pulse in the fully susceptible population into which the virus has been introduced, followed by somewhat lower levels of sustained transmission that sort of indicates sort of settling into a pattern of endemicity. However, we have had returns and spikes. So if you look at New York, where it first arrived, you can see that 2010 and 2012 were the highest years. Louisiana just had its worst season this past year, as did Texas, which had a monumental year of 1,700 cases plus. Idaho, it was there at very low levels for 4 years before it actually peaked with a big outbreak in 2006 where they got 1,000 cases. So there's a lot of variability as to what's going on there. It's not following any one single pattern. If we were to superimpose these on top of each other, so those are the same patterns now, but they're all on the same graph, you can see that the case levels also don't necessarily reflect the population sizes. Again, this peak right here is the Idaho peak, and that's not a very populous state, but they had nearly 1,000 cases, and California has never reached that level. New York has never reached that level. That's the red, the blackish line right there, and Florida has never reached that level, and it's the red line right there. So we're seeing a lot of variability in what happens, and what in fact is responsible for these differences and these different rates at which people are getting affected, at least based on population size, is not entirely clear, certainly from this initial viewing. There's also a lot of spatial variability. If you start to break it apart and look at the county level, you'll see that there's a lot of heterogeneity in what's going on. Very heterogeneous field there. Sometimes it's clustered around population centers. Sometimes, here's Mississippi right there, it really isn't. So the question is, well, what accounts for this variability? Why are we seeing some years that are really bad, some years aren't? Why are we seeing the spatial temporal variability that we're observing? So one of the things we have to think about is the force of transmission, okay? And this is a key determinant of whether there's going to be spillover transmission, okay? And one of the components of that is the number of infectious mosquitoes that are there, vector mosquitoes that are there, that are feeding on human hosts. So the idea there is that if we hold everything else constant, and we just increase the number of infectious mosquitoes, not the number of mosquitoes, but the portion of those mosquitoes that are actually infectious with West Nile virus, we would expect more spillover transmission, given everything else is equal, right? So one way that they quantify this, incidentally, in this force of infection, is through a designation or a measure that talks about vectorial capacity. And this is a measure of the pressure of transmission risk. There are a number of ways that this has been quantified. This is the one published by Garrett Jones in 1964. And he's got this equation where the vectorial capacity is equal to the vector-to-host ratio, which is m, times the number of hosts squared, times the probability of a vector surviving for a day to the nth power, where n is the time needed to complete that extrinsic incubation cycle, and then divided by another probability of vector survival. So the idea, again, is that, if you increase the number of hosts, or you increase the vector-to-host ratio, you're going to be increasing the vectorial capacity, okay? And the reason you're increasing the vectorial capacity, you can just think about in qualitative terms. If you're thinking of it in terms of just this transmission loop right here, this zoonotic cycle in which you're going to feed the virus between the mosquitoes and the avian hosts. If you increase the number of hosts, you have more opportunities to generate infections, okay? Furthermore, if you increased the ratio of vectors to that increased host population, you're certainly also going to be increasing the capacity or the number of bites that are going to be going on in the cycle. If you introduce the virus into that system then, where you have an increase in hosts and an increase of vectors that even exceeds that increase of hosts, you're going to get an amplification, as it's termed, of the virus within this cycle of transmission. So amplification is a term I'm going to bat around a little bit here. And what that is it's the epizootic cycling of a virus between mosquito vectors and avian amplification hosts. And that is what's considered necessary to achieve those mosquito infection rates that are sufficient to cause human epidemics. So when you have this epizoonosis, as it is, this epidemic within that zoonotic cycle of transmission between birds and mosquitoes, you're going to start to get a lot more infected birds, and you're going to start to get a lot more infectious mosquitoes. If you have more infectious mosquitoes, chance of one of those mosquitoes biting a human and being infectious is going to be increased, and you're going to get more spillover transmission. Now, the point when this is going to occur is when the vector mosquitoes and the avian hosts congregate together, when they actually come together in both space and time. That's what you need to do to bring this together, to elevate those two terms right there in the vectorial capacity equation. So to explore this, and this idea of co-location and whether this really is vital, we did some agent-based model simulations, and they in fact support the notion of it. So what this is an agent-based model tracks individual birds and individual mosquitoes flying around in a domain within it. And in that domain, we've set up some roosting sites where the birds like to congregate based on their patterns of going through their daily habits. And we've also set up some oviposition sites where the mosquitoes are going to congregate to lay their eggs, and then from there start to actually seek blood meals from that point on. Okay? So we can set up various configurations of it, and that's what this plot on the right shows you. We have 5 different roost configurations and 6 different oviposition location configurations. So there are 30 different combinations. And what it shows you is the fractions of each runs in which there was an amplification of the virus once it was introduced, or whether the virus dried off as the other alternative within this model simulated community. And what you can see here is the asterisks are the ones where the virus is introduced into an area where the mosquitoes and the birds are cohabitating or concentrating in space and time. And what you can see there is that those have much higher chances of actually undergoing an amplification in this model system than those in which that's not the case, okay, where they're not congregating in space and time. So this seems to be some model support for the idea that co-location is very critical. Some field evidence supporting this that was done here in New Haven, Yale, by Maria de Quasser, is that looking at what's actually going on in the late season and whether or not we can see changes in the estimates of West Nile virus infection rates within mosquito communities. And what they did indeed find was in the late season that there were higher rates of mosquito infection at communal roosting sites than at non-communal roosting sites. So that's another piece of evidence that there is, in fact, this co-location going on. All right, so what does any of this have to do with climate or weather or hydrology? Well, hydrologic variability influences mosquito abundance and distribution, okay? The first 3 stages of the mosquito life cycle, egg, pupae, and larvae, are aquatic. Mosquitoes depend critically on availability of water. Now, there are many different species of mosquitoes, and so they have different requirements. So we have to keep in mind and look specifically at the mosquito species that are critical to the transmission of West Nile virus, as the case may be. So rainfall has 2 principal influences on the mosquito life cycle. Firstly, the increased near-surface humidity associated with rainfall affects mosquito flight behavior, activity, and their host-seeking behavior. Some mosquitoes are very sensitive to this, some of them are not. But the ones that are sensitive, very often they'll wait and they'll hold their eggs and they won't actually become active until there's a big rainfall event. Near-surface humidity levels increase, then they'll go out and they'll look for some place to lay their eggs, and then they'll start host-seeking at that point. Rainfall also can alter the abundance and type of aquatic habitats that are available for the mosquito to deposit its eggs for oviposition and the subsequent development of immature stages. So we have rainfall as an input mechanism into those low-lying areas and the swamps and the river margins and the puddles and the ponds, the areas of water that the mosquitoes exploit for actual egg laying and where the eggs, larvae, and pupae go through their life cycle. So my advantage has been, well, if precipitation really isn't the thing that's guiding it, but it's feeding into these larval habitats, why don't we try to track those larval habitats a little bit more directly? So what we do is we use a hydrology model, dynamic hydrology model, to actually simulate the hydrologic cycle at the land surface and atmosphere interface right there. So there are a number of things that we can say about what's going on there using the hydrology model, a number of estimates we can make, but one of those is the estimate of soil moisture conditions or the water table depth, how saturated the soil is in a given area, in a given locality. And what that's going to give us is an estimate of how much water there is in the soil and how much of the lowland areas, the swamps, the lowland relief areas that are actually going to start to puddle and pond and potentially support the types of habitats that some mosquito species will exploit. And to show you why this is important, this just shows you some time series of precipitation. This is at a catchment outside New York City, actually near West Point. This shows you precipitation in a small catchment for one summer season. The red line is temperature. The green line is measurement of river runoff at the little weir that they have on this river. And then this is the model estimate of the depth of the water table, the point within the soil, the depth within the soil at which you actually get hit saturated conditions. It's a measure of the water content overall of the soil. And you can see that the way this varies, drying through the summer as the heat continues and then pulsing up if there's a large enough rainfall event, given how dry it is, the variability of this is very dissimilar from what you see going on in the precipitation record. So the idea there is if we can actually constrain this reasonably well, maybe this will give a better estimate of what's actually going on, and this is a more direct component of the environment to which the mosquito species might be responding. So within this hydrology model, not only are we including inputs of rainfall, but there are a number of other things that contribute to the energetics of the system, the drainage of water from an area, and its movement between the soil, vegetation, and the atmosphere. So I'm going to first talk about an example from South Florida where we've done. We're going to stay local, and we're going to consider it at a localized scale. We're going to be talking about two pathogens, West Nile virus and St. Louis encephalitis virus. We're going to look at it in a specific area, again, South Florida, and we're going to look at it in the context of the specific vector that transmits these pathogens in this area, which is Culex nigripalpus. So it's no longer this guy over here, but it's this one right here. So St. Louis encephalitis virus has a very similar transmission cycle as West Nile virus. As a matter of fact, when West Nile virus first came, they thought perhaps it was a new variant of St. Louis encephalitis virus. Before West Nile virus became all the rage and really supplanted it, you could see up to 3,000 clinical cases per year of St. Louis encephalitis virus. And again, the majority of cases are subclinical and mild, and there's no vaccine. The transmission cycle is really identical to what you're seeing for West Nile virus. Here's the Culex nigripalpus mosquito, the vector in South Florida. It's a ubiquitous breeder. It likes acid swamps, lime sinks, lakes, temporary pools, epiphytes. It'll breathe in a lot of locations, but it does have a preference, and its preference is for the puddles and ponds that develop on the Florida landscape after rainfall. It really likes the more fresh, rain-driven puddling that takes place that often can be filled with some eutrophic organic matter that makes it a little choked and fun to swim around and feed in. It feeds on both mammals and birds. And as a matter of fact, it has an interesting life cycle where it's been documented that it has a tendency to switch mid-season from first feeding on birds to later season feeding on mammals, which can make it a very efficient vector in the case of West Nile virus and St. Louis encephalitis virus. As I said, it's the principal vector in Florida. And one of the key things is that during the day, it will rest out of the sun in dense vegetation. And we'll come back to that in a moment. So there are four phases of the Florida SLE cycle in Florida, in this region. The first, broadly categorized in the first quarter of the year, is called maintenance. Amplification, that key component where you get that epizootic cycling between the birds and the mosquitoes, takes place in the second quarter of the year. And then we have two phases of transmission, early and late season, just to complete the quarterly designations. In 1990, they did have an outbreak of St. Louis encephalitis virus. There were 226 cases reported in Florida. Indian River County right here, where you get your grapefruits, was the epicenter. And over that span of time, they had a number of two pieces of data that they've been collecting through the course of this epidemic that were very helpful. The first was sentinel chicken transmission data. Those are the numbers right here, and this is where they posted sentinel chicken flocks. What they do is they take 6 chickens, they put them in cages out in a field someplace, and once a week they come along, they bleed them, and they look whether or not they've developed antibodies to the virus in question, in this case, St. Louis encephalitis virus. They've developed the antibodies. It's indicative that they've, in fact, been infected within the last 2 weeks or so. They'll pull that chicken out, and they'll put a fresh one in, and this way they get a time series for a specific location of the force of transmission in that area. Okay? The other thing that they have at this M right here was mosquito resting abundance data. As I said, they like to rest during the day in dense vegetation. Well, they went into a stand of dense vegetation there, and for 6 years, 3 times a week, they would go along with a vacuum aspirator and walk along a designated path and collect mosquitoes and take them back and count them and sort them by sex and grade, et cetera. So very rich data sets available for this time period. Some more specifics. Florida is a subtropical climate. Drought occurs seasonally during the spring, and summer is the wet season. Very different from much of the rest of the country where spring is wetter and summer is drier. Surface water is heavily managed, very flat landscape. There's lots of channelization in the area to help drain that flat landscape so you don't get too much flooding and ponding all over the place. And also, the landscape is fragmented. So this is what one of those hammocks looks like where the mosquitoes might be resting. This is an area of lush subtropical vegetation. But those only dot the landscape at this point. There's been enough development in Florida for housing, for agriculture, that you have most of that cleared away, and then you'll have little pockets between those where you have this lush subtropical vegetation that the mosquitoes like. So what we did is we modeled the hydrology for this area. We used this model, what we refer to as the top model-based hydrology model. It's a bucket model, basically. It discretizes the soil into 10 layers, and then you track the movement of water and energy between those layers and between the surface layer, vegetation, and the atmosphere. It's really run as a boundary layer problem. It's forced by hourly surface meteorological conditions, things like precipitation, temperature, pressure, wind speed, humidity, et cetera. In addition, it also takes into account topography, which helps control rates of runoff, even in the Florida landscape. What we can do with that, by integrating it forward in time, is we can produce a time series of the estimate from the model of the mean water table depth for the area that we're looking at. So it's a relative measure of how dry or wet the soil is and how much puddling and ponding might be supported by that soil at a given time. So that's the blue line right here. And you can see this over a 6-year period from January 86 to the end of 1991. It shows the variability in hydrologic conditions as estimated by the hydrology model. You can see there are peaks right here in the summer, and there are low periods right here, which is in spring taking place. Okay? In addition, on here, I've shown you the data from those 5 sentinel chicken sites, and it shows you the weekly rates of seroconversion of the sentinel chickens. And you can see that there were only 3 seasons of the 6 in which there was any seroconversion: 1986, 1989, and 1990. And in all 3 instances, it occurred in the season after a period of drought and during a period of wetting, with the big one taking place right here. In addition, you can see that they all occurred actually after the 3 driest seasons. Okay? So we can do some analysis of this, and we subjected it to a bunch of statistical analysis and show that, in fact, this association is significant in that drought 4 months prior and wetting half a month to a month prior is significantly associated with the seroconversion of those chickens here. Okay? So we see this transmission occurring after a period of drought and immediately following a wetting period. So the question is, well, why? And the basis of the relationship really came down to the mosquito collection data taken in this hammock. What these plots show you is each of the 6 years during which this was taken. 89 and particularly 90 were the 2 really bad years in terms of the seroconversion to sentinel chickens, and 1990 was the case that was broad epidemic across much of Florida. And the plots show the total number of females collected as a function of modeled water table depth the same day, measure of the drought. Okay, and what you can see is that you get a general increase as it gets wetter in the numbers. It's very loose and fuzzy there, a lot of scatter and noise there. But on the 2 very dry years, 1989 and 1990, there's a bit of a bimodality. At the very driest conditions, you can see there's an increase in the number of mosquitoes that are being collected. So there really are 2 interpretations for that. And one is that mosquito abundance actually increases during very dry conditions. Well, the latter is that the mosquitoes are congregating in the hammocks during drought. And it's actually the latter. Lots of observational evidence, lots of anecdotal evidence. I've been down there in droughts. I was down in the Everglades in the drought in 2004, and people are coming along and they're looking at the alligators there, and they're sort of having a shrinking habitat, the alligators. And there are also these paths that go into these hammock stands, lush native vegetation. And I was just standing outside and I'd watch people walk in, and 30 seconds later they'd come running out because of the mosquitoes. And the mosquitoes are really active in there, and they're all in there. You stand outside them, you don't get bit. You go in there, you get eaten alive. I tried it myself once. It wasn't fun. So they're there. So what do we think's going on? Well, spring is the nesting season, and the birds are exploiting those hammocks as well. So remember, a lot of the Florida landscape, particularly in Indian River County, has been cleared. The birds really exploit that lush subtropical vegetation that's dotting the landscape, and it's those same sites that the mosquitoes are retreating to, okay? What do we have? We have hydrologically induced co-location of the avian hosts and the vector mosquitoes. We get amplification. So that's what we think is going on in the springtime there. Since then, we've looked at this a number of different ways. We've looked at the transmission of St. Louis encephalitis virus to wild birds. We've looked at high levels of it, epidemic levels to transmission to sentinel chickens throughout the state, not just in Indian River County. We've looked at human cases throughout South Florida. We've looked at human cases of West Nile virus throughout South Florida. They all present the same relationship. Drought followed by a wetting leads to an increased likelihood that you're going to see human cases of West Nile virus, elevated levels of sentinel chicken seroconversion, cases of SLEV, et cetera. So our conclusion hypothesis is that spring drought restricts the nigra activity to select refuges, those hammocks, where nestling, juvenile, and adult birds are found. And that forced cohabitation of the vector and the avian host permits that rapid amplification of West Nile virus and St. Louis encephalitis virus. When the drought ends, the infected mosquitoes and birds disperse. And they come into broader contact with humans. They start entering the agricultural plantations. They start entering the housing developments. There's more interaction and contact with humans, and you get this, you get transmission to incidental hosts. So we've since taken this and tried to look at some other places. Again, they have different vector species, different avian host compositions. They have different climates, different land use practices in them. But looking at eastern Colorado, we found a very robust association that was the opposite. It was wet springs and dry summers favoring human West Nile virus transmission. Okay? So what could possibly be going on? Well, we have to think about the vectors there and what their specific interests are. And what we're speculating at this point, we don't have as much data there. We don't have a rich set of entomological data to look at, for instance, or the sentinel chicken data yet, is that we speculate that increased land surface wetness produces a greater abundance of mosquito breeding habitats. So if they get a late winter snowfall that's particularly heavy, you can keep the ground very wet. You can get a lot of spring melt. And Tarsalis in particular likes to take advantage of those types of breeding habitats. And you can get a big bloom in those numbers. Then when you have a dry summer, you're losing all those water resources. And the birds and the mosquitoes have to congregate around those limited water resources. And that can put them into sympatry, and you can get the amplification of the virus in that instance. I think the Culex pipiens particularly exploits that latter dry summer mechanisms because it really likes eutrophic larval habitats. We also think that flood irrigation in eastern Colorado is a very important confounder in this that's actually amplifying the amplification problem because they'll take the limited amount of water out of the river system and they'll use it to irrigate a lot of croplands. And it's not necessarily done in a way that's not conducive for producing certain floodings of the fields. If that's the case, you get problems there also. Also see a lot of transmission in Colorado around golf courses in the Fort Collins area, for instance, where they have water traps. That's where the mosquitoes and the birds will hang out together. We've also done and extended this and looked at Long Island, New York. And in this case, we've been looking at pooled mosquito infection rates. We've compared that. We get the exact same relationship we see in Colorado. And again, the climate of Colorado and New York is a little bit similar in the sense that they both tend to have wetter springs and drier summers. We're seeing sort of a similar incidence there. So one of the things that we want to do is we want to look elsewhere. We want to start to try to put this mosaic together by looking at hydrology and these incidents of West Nile virus, the dynamics of it, whether or not there's this hydrologically induced forced co-location of birds and mosquitoes, and that's consistent with the emergence of transmission episodes in space and time. So we can do this from a number of advantages. We can use both model simulations and satellite observations of hydrology to make these, to do this work. And ultimately, the idea would be to try to monitor the whole country, potentially in real time, and see what's going on. In the meantime, we have set up a real-time monitoring and forecasting system in Florida. So here we're using this NOAA-NASA National Land Data Assimilation System project data to force a hydrology model and produce simulations of hydrology in Florida. They'll look something like this when you do the runs right there, and this is some of the input data that goes into the model at an hourly clip. And what we do with this observational use is we use it to actually inform local county control officials and public health officials about what's going on. The relationship in Florida is uniquely so robust, at least uniquely at this point, that we're actually almost able to take the vector mosquito out of the equation. And we can just look at hydrology and use it as a determinant of risk for West Nile virus at this point. And that, in some sense, is what we're doing. And we've been able to alert local county officials when the drought conditions have been consistent and conducive with the type of dry down that you see that permits the amplification of the virus, when the timing is right. And the results are kind of interesting in the sense that obviously, if they take control measures, it's hard to know whether what we predicted would have happened because they've done something to counteract it. And Florida has yet to have a large outbreak of West Nile virus, which is curious also. A very large elderly population. You would expect that to be a little bit more susceptible to it, and there would be more problems with it, but it has not happened yet. Where they have had outbreaks have been in small communities, and they've been very focal, and they tend to be wealthy communities in urban areas. And what they are is they're people who are essentially, because they're so wealthy, they live inside a hammock. They live inside an area where they have all that lush subtropical vegetation, because it's great and it's beautiful. And basically, within those little islands, like Coconut Grove, Miami, the mosquitoes and the birds find their paradise, and they come together when it's drought, so they don't need the wetting to take place. So, in fact, we haven't had a really good episode of drought and wetting as occurred in 1990 since West Nile virus appeared in Florida. We've just had drought that's occurred, and then it just continues to dry. They've been having a lot of problems with drought in Florida. So the drought is sufficient to get the amplification going in these hot spots within the hammocks, but if the mosquitoes don't have that cue, that wetting to disperse, they're not going to infect a lot of people. The exception will be for people who happen to live in hammocks, and that happens to be the very wealthy areas in Tampa and Miami, et cetera. And that's where we've seen the transmission. So some of the other issues that we're trying to think about is, well, how do we translate this? How do we operationally use this? We're making probabilistic forecasts of what's going on, and how do we use this information that only really provides likelihoods to communicate to public health people? And that's something that we've been trying to work on a bit, and trying to sort of engender an understanding of what their needs are and also how to better communicate it. And in fact, in truth, providing probabilistic forecasts is a much richer palette of information, and it's a way to go. Rather than claiming that there will be this event, which you're going to be wrong sometime, you want to get them a distribution of outcomes and an understanding that there are chances of certain events taking place, and there may not be in other instances. Okay? So the last thing, of course, is that while it'd be nice to really show that the monitoring system anticipates that huge epidemic that we think it might happen in Florida if conditions are right, it would be really nice if we could actually avert that epidemic by taking some proactive measures to counter it. But then the question really is, how much should you do? Should there really be incredibly heightened targeted control of vector mosquitoes, which they don't do? I mean, they will spray more, but should we tell them, go into the hammocks, try to clip it really early on in the spring? You have a drought going on, try to nip the amplification in the bud, as opposed to waiting till the transmission season starts to tell them to really become proactive. So we need to look at this. This requires some more modeling, even some economic analysis to understand. So in summary, we found that amplification and transmission of West Nile virus are intrinsically linked to hydrologic variability, and this association varies from place to place as the vector mosquito and avian host composition changes, as well as land use practices, as well as hydrologic and climatic conditions of a region. Okay? But if we can account for those differences, we should be able to identify the different links potentially between hydrology and West Nile virus dynamics, and that much of this link really comes about aggregation or concentration around limited water resources. At least that's what we hypothesize. Right now, we are doing real-time monitoring and forecasting, and they are possible, and it's in operation in South Florida. And so just to conclude, these are my collaborators, include Jonathan Day, medical entomologist at the University of Florida, Mark Stiglitz, who's a hydrologist at Georgia Tech, and sundry other people, Scott Campbell, Mark Kane, Gary Harding, Nick Comar, and Steve Zebiak. Thanks.